Good morning, good afternoon, and good evening, everyone, wherever you may be in the world. A warm welcome to our presenters and all of you to another Marine Biodiversity Networking Friday. This session is part of a webinar series on GOOS BioEco Essential Ocean Variables. My name is Dr. Sean Porter from the Oceanographic Research Institute in South Africa, and I'm a coral reef ecologist and will be moderating this session. Before we get further into proceedings, I need to highlight a change in the program. Regrettably, due to unforeseen circumstances at the 11th hour that are out of our control, we are having to postpone the component on coral cover and composition. So this session will therefore focus on microbe biomass and diversity only. We apologize for this late change. As many of you here will appreciate, microbes are essential for ocean health, forming the base of marine feed webs and mediating biogeochemical cycling. However, the impacts of warming, acidifying, and deoxygenated oceans on key microbial functions, such as carbon sequestration, vitamin and nutrient provision, are not well understood. The Microbe Biomass and Diversity Essential Ocean Variable outlines protocols and data portals to help address this challenge. This webinar will therefore dive into these aspects with examples underscoring the impacts of large-scale microbial observations and what can be achieved with coordinated efforts. We are fortunate to be joined by two experts in this field, Dr. Alejandra Prieto-Devo from the National Autonomous University of Mexico and Dr. Julie Robidart from the National Oceanography Center in the United Kingdom. Dr. Alejandra Prieto-Devo is an oceanographer and full professor in the Department of Chemistry at UNAM Sissel Campus, Faculty of Chemistry. She holds a PhD in oceanography and a master's in coastal oceanography. For over 15 years, Dr. Prieto-Devo has investigated marine microbial diversity in coastal ecosystems across Mexico, including studies in coastal cenotes of the Yucatan Peninsula. Since June 2024, she has co-led the Microbes, Biomass and Diversity Essential Ocean Variable Panel within GOOS, thereby contributing to global efforts in ocean conservation. Then it is my pleasure to introduce our second speaker, Dr. Julie Robidart, who is the head of Ocean Technology and Engineering at the National Oceanography Center in Southampton. She has been working on marine omics for 25 years in the US, Australia, and UK. She's an expert on marine microbiomes and their mediation of carbon and nitrogen cycles. Her work centers around scaling marine observations, developing a suite of new deployable technologies to broaden the time, space, and taxonomic coverage for marine organisms. Welcome, Julie and Alejandra. Lastly, before I hand over to our speakers, some housekeeping. Each speaker will be given roughly 15 minutes to present. After both speakers have presented, we will have a 15-minute questions and answer session. As such, please post your questions in the Q&A box rather than in the chat. Once the speakers have finished, I will read the questions out to the speakers for further discussion. Okay, I think that is all for now. Please join me in welcoming our first speaker, Dr. Alejandra Prieto-Devo. Over to you.
Thank you, Sean. Thanks for the introduction, and hello, everybody. Julie and I are very happy to be here to show you what we've been working on in the past few months in respect to the microbe spec sheet from GOOS. I'm going to try to find the presentation. I'm sorry, I had it right there, and now I can't find it, but give me 1 second. Here we go. There. So I'm hoping that everybody can see it. If not, please raise your hand or something, but can you see
Yes.
it? Thank you. Thank you, Sean. Okay, so today we're here to tell you a little bit about GOOS, the Global Ocean Observing System, which is an effort uh, that is directed by UNESCO, a UN effort. So it started in about 30 years ago, 1991, as part of an intergovernmental oceanographic council. So, so what we're gonna tell you is how GOOS came. Well, this is my part, like how why this was created, why this effort has been created, and how we've been contributing to it through the Microbe spec sheet. And unfortunately, as Sean mentioned, we won't have the coral cover and composition today. But the if you do corals and you want to stay and see how the spec sheets are divided, the sections of the spec sheets are usually the same. It's what's in each section that changes a little bit regarding on which oceanographic variable we're talking about. So basically, what I want to talk to you about is how we have joined the effort of observing the ocean, right? And everybody in different parts of the world is doing oceanographic observations and research. But the GOOS initiative is actually it was created for. It was so that we can all join together and give information in a cooperation, right? And in a similar way, so that we can all have all of the data, that we can get access to all of the data, so that we can create models and we can take actions toward policy, so that the people that make the policy have the information to take action into what's happening in the ocean. So it refers to how the communities adapt, what the health of the ocean is, how you can take climate action regarding this, and other aspects, right? So this started as a more of a physical oceanography initiative, where a lot of people started observing all the phenomena in the ocean, and they started gathering all the information all around the oceans in the planet, and then they decided to coordinate and say, okay, now if everybody joins and we can put all of this information together, then we can create models and we can see what's going on. And then as the physical oceanography part started, they started realizing that the ocean is a whole, right? If you're an oceanographer like me, you know that everything is interconnected. So if you study the ocean, you know that everything affects whatever you're studying. So then they started gathering more people that are actually studying other parts of the ocean, right? So this is what basically what GOOS does, is it coordinates observing, let's call it like observatories, so that they can create through them you can create partnerships and then get the data to the different observatories, and then that will in turn be it will definitely help users and other maybe policymakers to take action, right? So the vision is to really create a huge all over the world observation of the ocean and integrate it, and then be able to make decisions and to see what's happening to the ocean with what we're doing in the world, right? All the anthropogenic influences and how they affect the ocean. And as part of the UN, it tries to do it all around the world, and to basically GOOS tries to create a community to make these partnerships happen, right? So a way to access all of the information for everybody to be able to see it and to use it towards whatever it is they need. So they're basically, it's the creation of this network, and then you need a place to put all of this data, and that's what OBIS and ODIS is all about, and we will talk a little bit about that, so that you can use that data to create information, so that you can either do warning systems like what we do with climate when there's a hurricane, or when you need to control certain things in the ocean, like, I don't know, things to do with fisheries, for example, and basically to plan what you're going to do inside the ocean, because we want to preserve the functioning of the ocean. So what is an essential ocean variable? Very quickly, it's the minimum set of variables needed to assess ocean state and variability that are important in a global state. So what is it that we need to observe about the ocean when it relates, for example, to climate? An EOV would be related to physics and climate. If operational services would be biology and ecosystems, and ocean health, for example, like variables related to biogeochemistry. So we are going to talk about the biology and ecosystems EOV today, and then we have subvariables, which are so these are the three main variables in the ocean, and then the subvariables, which is each of the little ones we talked about. We have, for example, the basic measurements to what you need to know about biology and ecosystems regarding the organism that you are studying, or the area that you are studying. And supporting variables usually are interchangeable between these EOVs, and it's basically the anything that surrounds that ecosystem. And then from there you can get like derived products. So that means that what you use from all of this information to actually set out and say this is what's happening, interpretation of that data. So if we all gather together and we do this in a coordinated manner, and then we know that there's people doing it in one place, there's people doing it in another, but we have the same basic information about whatever it is that we're studying. In this case, we're going to talk about microbes. So if we know the basic information that we need about microbes to put into a data set so that it can be used to obtain information that coordinates with the rest of the variables. So I'll show you. So this is how we want to compare ocean data from all over the world. So if you do that, then you can use that to give information towards ocean health, which is like the main variable. Remember, I said it was the first three EOVs: the ocean health, biology and ecosystems, how and what is the state of the ocean, right? Like ocean health, biology and ecosystems, which would be operational, and the physical part would be how it affects climate, or how climate affects your variable, more likely. So what you want is really to have like sustained ocean observations. And if we're doing observations of the ocean in different places of the world, and we can do it continuously, and then we can get the data from those informations to be shared to everybody around the world. So let's say you go out and you you collect your information, you publish your paper, or you just have the data there because you don't have time to publish it, but how to get it out so that people can use it, and it can be part of a bigger thing that can be used to actually give you more from the data, right? So we have the functional groups of these EOVs. There's specification sheets or spec sheets for phytoplankton, so plankton, seabirds. The ones that are in dark green, it's because the specification sheets have already been published, or they have been finished. And then we're working on the benthic invertebrates, and the microbe one, it's still in process, right? And then you can actually use some of those to talk about variables, to talk about the state of the habitat or the ecosystem, like seagrass cover and composition, macroalgal canopies, mangroves, and coral, right? And then there's cross-panel EOVs, like I mentioned. There are things from the physical oceanography EOVs that affect the biology and ecology EOVs. So, but if we have the same structure on how we get the data, and we have it available, then we can use all that to actually start creating policy, or start making or getting people in power aware of what's happening in the ocean. So basically what GOOS is trying to do is provide the framework for coordinating the ocean observations so that everything is comparable, right? So it doesn't mean that everybody has to do the same thing. It just means that when you share it, it should have like a same structure of how you share it, so that the computers can read it, basically. And this is where OVs and ODs start working. And the coordination of everything, right, is because you get a format on how you're sharing your data, and that way it gives information to a computer that can extract that data and use it accordingly. Because if we just have everything explained in different ways to the computer, then the computers will go crazy. I mean, we do have AI, but it doesn't work still to like understand everybody's way of collecting data. So this is what basically the GOOS organization or the GOOS effort is to try to get everything into the same format to present it to a computer, basically. So I think that's what I wanted to tell you about GOOS, and Julie is going to take over to talk about what we've been working on the microbial biomass and diversity spec sheet.
Great. Thanks for that, Alejandra, and thank you, Sean, for the introduction. Yeah, so this is, as Ale said, it's like an evolving EOV spec sheet. So this is the first draft of it. That means we put some work into it in the last year. We've gotten a bit of feedback, but we're open to more, and it's not yet final. I think it's going to be a living document. So as we go through, if you want to give us feedback, please contribute. So I wanted to start with an introduction into what can be done. So if we were to do what Alejandra said and get all of our data into the same database, we can, in theory, we could map it, but we could also do a lot more and learn a lot more about our interconnection ocean. So it's not that the Indian Ocean is separate from the Atlantic Ocean. The Indian Ocean feeds into the Atlantic Ocean. And so in order to understand how your regional observations that are giving you your science, answering your science questions and giving you your data sets, how those connect to other oceans and how they'll change over space and time, it requires a lot more coordination than we currently have. So a really nice, actually go back to that, Alejandra, if you don't mind. A really nice kind of impact study that demonstrates this very clearly was this major Tara Oceans expedition. So you can see on the left all the different stations that were stopped out with the Tara schooner. And this resulted in this Science Special Journal publication with a bunch of different insights into marine microbes, which you see on the publications on the right. So we learned that most eukaryotic biodiversity is from uncultured heterotrophic single-celled protists, which is very different from how we think about it. We usually think about eukaryotic phytoplankton. There was a big, when you put all the data together, it looks like temperature was a major driver for community composition. Viruses were looked to have they're transported with ocean currents, but it looks like regional structure has to do with their ecology. So they diversify based on local impacts. So these are major things that we found out because there was this coordinated effort with everybody getting their data together. And as we think about these major problems, like Alejandra we're talking about global change. The next one's fine, Alejandra. We're talking about marine heat waves and ocean acidification and deoxygenation. You have this interconnected ocean, these major interconnected problems. And if you do larger scale observations and put all those data together, you end up being able to say a lot more about the connection between those changing ocean conditions and the biology of the ocean. So this on the right is a study from the CPR survey in the North Atlantic, which is this really high density survey that basically showed as changing oceans in the subpolar and subtropical North Atlantic change, they have different effects on diatoms. And diatoms, they're important for carbon sequestration and food webs. But this kind of information is really valuable for understanding how changing ocean regimes are going to impact the biodiversity of the marine environment. Yeah, go for the next one. Thank you. So for this work, we had to restrict our definition of microbes. So this you might recognize as the microbial tree of life. It's based off of metagenomes. and it contains the bacteria, archaea, and eukaryotes. And in addition, this candidate phyloradiation that was kind of discovered when all these metagenomes were put together. This specific tree introduced a lot more diversity than we knew existed previously. Microbes have been evolving for longer than eukaryotes, of course, so they have a lot more diversity. And for the sake of the GOOS spec sheet, we decided to not address the phytoplankton as in depth because Frank Miller-Karger and Rafael Crudela already have a spec sheet on phytoplankton, which you could hear about at the next Air Center webinar. So we restricted our microbes definition for the spec sheet to be microbes that are not phytoplankton, and they touch on viruses, the spec sheet, but it's not really in depth on viruses either. There are a few protocols in there, and it's addressed in one of the phenomena, but it's not really in depth. So with all of that, we're looking at organisms that don't necessarily have a morphological distinction, right? So you can do microscopy, but microscopy alone, without a probe or something, it might not tell you much about what you're looking at. So a lot of our tools have to do with sequences, because the sequence data, the genomes themselves, have a lot of diversity that we can't necessarily capture using other tools. And when we think of the information that you would gather, typically as microbiologists, we think about the derived products. So we're thinking about microbial community composition, microbial function, phylogeny, these kinds of things. But what those are a combination of our subvariables. So the two subvariables that you would use in combination are the concentration of the microbes, so your abundance or your biomass, and the diversity, so who's there and how many are there, basically. Yeah, so you could collect those derived products from that, and then is there an animation on this, Ale? Yeah, perfect. The next thing you would want to know is the environmental context. So in order to relate that organism to their environment, you want to know what's the temperature, what depth are they at, what's the salinity, and probably something about the chemistry. That lets you it allows you to do a bit more with those data, kind of see what's driving the community structure, but it also allows you to, in theory, model where they might be happy to live. And in addition, there's EOV-related supporting variables. Those are things that have to do with the EOV itself. What's the size of the organism? What's the functional diversity, growth rate, that kind of thing. And while we couldn't be comprehensive with the specification sheet, it's supposed to give you an idea of how you would go about looking at different phenomena. So we chose 4 phenomena for this specification sheet. We could have chose 100, but these have to do with, so the first one, geographic variation in diversity and composition, that's kind of biodiversity, that's oceanography as we usually think of it with microbes. Roles in transport and cycling of elements, that's around biogeochemistry. And then there's a couple that have to do with pollution. One's around fecal indicator bacteria. Those are methods that have to do with how to look at sewage spillage. And then the other one is around oil degradation by microbes. So if you look at this little figure on the right, what we're looking at here are biogeochemical cycles, your biodiversity, and then 2 that are related to pollution. Even though they have in this figure, as you can see, a lot of diverse roles.
Just to highlight a little bit, in case you're studying anything related to this EOV, if you're studying microbes, it doesn't have to be, just to remind you, it doesn't have to be these 4 things that you're studying to fill out an EOV. This is using these examples what you would need to fill out the boxes that we have in this specification sheet. So this is just like a guide you can be studying any kind of phenomena related to microbes in the ocean. And then this is the kind of things that would be needed to be addressed in each of these boxes. That's all.
That's right. These are examples, basically. Yeah. And related to each of those, so what we're looking at now are the specifications for those specific phenomena. So yeah, you could see that this one is around the oceanography type phenomena. So what you could see are we've linked in some references, and those are linked protocols or frameworks, guidelines for some of the programs that measure that. So for example, with here, you've got geographic variation in diversity and composition. You can also look at the role in transport and cycling of elements, so biogeochemistry, with these guidelines that are used in these ocean time series programs that are linked on the right. And it doesn't have to be like Alejandra said, it's again not this is not restrictive. You can do what you like. These are guidelines for what the community follows in case you need some ideas for kind of shaping your sampling program.
Right.
And similar, sorry, go ahead.
No, I was going to say, because you mentioned that this is very like oceanographic like, right? And this is because, as I mentioned in the introduction, it's built on from all of the specification sheets that we had for the physical and climate EOVs, and that's how it started changing. So that way we have the same kind of sections, let's put it that way, so that they're comparable, and they can use the physical oceanography EOVs to see how everything integrates into the models.
Right. And yes, and it's similar. The one on the right is around fecal indicator bacteria, but it's the same structure, like Alejandra said, here for the different phenomena. And yeah, and then here again, these are links to the specifics of the protocols. So your approach might be cultivation dependent or bacterial counts, or it might be looking at DNA. So each of those approaches, we have some suggested methods and best practices that are available and are linked in the specification sheet if you're looking for a new protocol, let's say. And do you have to use those protocols? No. So especially when we're thinking about DNA, there are a lot of different ways that people approach the analysis of DNA. You could look at specific genes, or you could look at the entire genome. You could look at RNA if you're interested in activity. But what we're aiming to do is kind of collect the data, put them together, but also there's a lot of efforts underway already that are trying to compare the different protocols. So these intercomparison exercises are international. There's these big international groups that get together and compare things like filtering with just a Sterivex and versus filtering with a bunch of different pore sizes in a size fractionated sampling. And in addition, on the bottom left, there's a bunch of different eDNA auto-samplers, and these are used for microbes as well. And there's some intercomparison exercises underway already that are kind of comparing how the data look when you sample across all these different technologies. So the idea is, it doesn't have to be exact standards, but it does also help when you have the capacity to compare your protocols to the existing protocols. So that's the sampling part of the workflow. On the right is kind of a bit around the Glomicon effort. That's around kind of the extraction, and the sequencing methodologies. And then there's the pipelines used. So there's a lot of diversity that has been compared, and that still remains to be compared through ring testing and things like this. And as these evolve, the specification sheet can be updated with that information, which is really helpful. So we can, if you have a standard operating procedure that you use, it would be really useful to get it on the ocean best practices system that kind of it tells us that it's been reviewed, and but intercomparison exercises as well. All of this information basically will be linked to the specification sheet as it rolls out. And this is the most important bit. So this is what Alejandro was saying is, you have your regional study, you've got this data, and you can publish your data. But the best thing you could do to get the most value from it is also, and for funders and the people who are interested in supporting your work, is to get those data online and available. So you can already submit the metadata to ODIS, the Ocean Data and Information System. And this would be your metadata, like we talked about the subvariables and the variables, and it would basically be what's the latitude, longitude, what were the conditions, what was the depth, etc. And that ODIS goes back and forth with the OBIS, and so OBIS is what Alejandro mentioned previously, but you can basically publish those data together, and it'll get basically if you put it in ODIS, it'll get to OBIS. There's a BioEco portal specifically that when you flag an EOV, it will be linked together with the other data from that EOV. So Alejandro, yeah, you want to talk about this?
Sure. So recently we, a bunch of people that do microbial ecology got together, and we started looking at how many microbial observatories we could name. And these are some that we could come up with in the list. So these are all observatories that have been published, that are out there, and where people do constant sampling on the ocean for microbes, right? So there's at least 30 of these observatories. So it would be great if any of you start creating an observatory or already know about one that's not here. I just learned yesterday that they're creating one in Chile, and so it will hopefully like soon get into this list. But that's another thing that's important to know, that there's already a lot of people doing this work. And just as Julie mentioned, the whole effort that GOOS and the United Nations are doing through UNESCO is trying to get all of this data from all of these observatories to be interconnected with everything else that is observed from the ocean. So if you do microbes, you can, if you put it in OBIS with a B, then like when you put a notice, ODIS immediately transfers it to OBIS, and so it's available for OBIS, and then OBIS can use that information to relate it to everything else in the climate and physical oceanography sector, or in the geochemistry, biogeochemistry EOV. So that's the important thing about this, is that it will be interconnected. So that's why it's important to actually put your data out there. And if you have the data, there's ways that you can actually publish the data as a paper without having done any work on it yet, but publishing your data and all of the methods that you used, which is very, very important. What are the methods that you used to gather all of that data? What filters did you use? Did you pre-filter or didn't pre-filter? If you yeah, I don't know what else. If you got like, first, what kind of other variables are very important to have for the metadata? You can see all that, what ODIS is asking for, and then you can plan so that your data has all of this information, and then you can put it in. And as I was saying, you can publish the data in journals that dedicate themselves just to publish data. You don't need to have done anything on that data. And then once it's publicly available, you can continue your research. If somebody else uses the data, they will cite you because you have a publication on how you got that data. So you will not lose your data to somebody else, which I know it's something that worries people a lot. But we need to start creating ways of sharing the data without people just taking it from you. And that is one good way of doing it. Look for those journals that actually do that.
Yeah. And yeah,
Oh, no. You...
visible disk.
Sorry, I think you're cutting, but I think what...
Yeah, so
what...
Oh, yeah.
Go ahead. Go ahead.
I just said in the chat, Elizabeth just corrected us that if you publish with OBIS, it gets visible with ODIS. So we said that
so it's the other
backwards,
way around. We said
Sorry.
it backwards. Okay, sorry about that. Thank you.
Yeah. And I just wanted to, because Alejandra had that large list of observatories, and basically I wanted to end on the note that if all of these were together in a map, it would be fantastic to be able to look at them, all the data together, right? You would have a nice global coverage. What you're looking at here on the left are the metadata that already exist for microbes in OBIS. And on the right are the data themselves. So it's the different sites and collections that have uploaded their actual data to OBIS. And if you look at Australia on the bottom right, you could see that you could see the national reference stations for Australia very clearly. And yeah, it means that those very specific sites, just like this, and that's one of the observatories that was listed previously. They've all been able to get their data in the right format and uploaded to OBIS. It's lovely.
Yes.
Yeah. Yeah. So that's the vision of it. And I guess Alejandra, did you have anything else to say?
No, actually, that's perfect. Yeah, I just wanted to say that. Oh, well, maybe when you look at this, one thing, when you create these maps and when you see where the data is and what has been done, then you can find the gaps. And that's more like, it's very important to know where things have been done, but it's also very, very important to know where we need to go because we're missing the data, right? And as you can see for microbes, there's still a lot of work to be done.
Right. Thank you very much, Alejandra and Julie, for a most insightful presentation. It is impressive to have an overview of what GOOS aims to do and how microbes fit into the bigger picture. We will now move on to the question and answer sessions. I see there's 3 questions. I'll just go through them systematically. So the first question is from Balaranwan Balugan. And the question is, the ocean is vast and no one country can study it alone. What is GOOS doing to ensure all the nations in the ocean basin are involved in generating the EOVs to add to the global system? Are there specific painstaking efforts to carry everyone along? I'm not too sure which one of you would like to try and tackle that question.
Right. I think this is part of the GOOS effort precisely. It's because nobody can do it alone. So the nations in the ocean basin are involved. That's a very good question. There's definitely a big effort to get people involved from all over the world into all of the GOOS panels. There's usually calls out for experts on the area. So I think there's actually going to be a call soon for one of the EOVs. I don't remember which one, but they're constantly trying to get people into the group of people that sit to discuss all of this information that will be shared. The most efforts that have been done is when we all get together and we try to discuss this as a group, and we try to, like, all of the BioEco panel gets together, and then you start talking about what is important for all of the biology and ecology subvariables, what is important and how to get it out there, and then how to spread the word. Basically, and these webinars are precisely for that, is to tell people what the effort is, to also tell you that to be aware that this effort is out there, and then there's constant calls for people to join the panel, because this is I believe it's every 2 years, right, Julie? Maybe you remember better. Like every 2 years, there's a call for people. So we send out, we put it out in the webpage for GOOS, and it's also out from our colleagues. We start spreading the word so that you can apply to be in the group. And what they're trying to do is trying to cover precisely all of the areas of the world, so that as you can see, like many areas of the world don't have the same opportunities or means to do the research the way maybe the global north does it, but it's still very useful information. If you don't have enough resources to do sequencing of everything you do, but you have viral and bacterial counts, that's important information, and it's important for you to put it out there, because you're still observing microbes in the ocean and you're still getting data. So it's kind of that, bringing everybody together and getting the word out. And as I said, the IOC started in 1991, but this part of the EOVs, and specifically the microbes, it just started a couple of years ago. So we need to spread the word and have more people come into being part of because we coordinate the efforts maybe, but we have workshops. For example, there's a workshop coming in November where we contacted all of the people from different basins, ocean basins. We contacted people from Japan, from Korea, from South Africa, from everywhere that we knew we could find someone that works on microbes, and then we asked them to join the workshop so that we can fine-tune the spec sheet. So those are the kinds of efforts that GOOS is coordinating. Unfortunately, all of this, to do it in person, always depends on funding, and as you would understand, there's not a lot. So sometimes it's the meetings where all of the panel gather, they're very localized, because then people in Europe can go, or people in the South, wherever it's in South America, for example. If you do a panel in Brazil or in Argentina, well, probably more local people will join. But that's the idea, is to do all of these things in various different regions of the world for the meeting, so that everybody can join.
And on the data acquisition side, there is a protocol, and now the marine genetic resources. There are some things that the scientists should follow when they're collecting samples and doing science. And so I don't think OBIS currently has a checklist for that. I think that's kind of a little binding thing with the different nations that exists, but yeah, not at the moment. It isn't necessarily confirmed when those data are put into the system. Maybe Elizabeth knows more about that. But I would say philanthropy are doing well, they're interested, I think, in bridging these gaps, and also getting them filled with folks in country that would be able to sustain observing programs as well. So there are some efforts that I'm aware of with the Westpac region that are kind of underway to get scientists there funded to do some research, to fund international research, but philanthropy are one of the means of doing it. So.
Thank you, Julia and Alejandra. Maybe Julie, you can take the next question. It's from David Pelisek. Can you comment on interconnection between genomic diversity in EOV and EBVs in terms of community coordination? Yep. Where should microbial metagenomics person contribute?
Both. So there, ultimately the metagenomics is telling you a bit about function, isn't it? A bit about biodiversity, but it is a measure of the diversity EOV. And when we talk about community composition, when it derives products, that often brings in a lot of metagenomic data. But those are complementary, the EBVs. The EOVs complementary, but don't necessarily stand independent of one another. There is a lot of overlap. So yeah, where can you contribute as in contribute your data? I think we are all coming together around getting data into OBIS, both EOVs and EBVs. And I don't think you don't necessarily have to exclude one. You can be involved in both. If that's What do you mean where
Yeah.
we can contribute?
I guess maybe it depends on if you're using your metagenome to describe something about the ocean, right? If you're just, it depends if you're just looking at if it's an exploratory study, then you would just use the information in your metagenome for, as Julie mentioned, for biodiversity, and to say, okay, this is what's here in the ocean, and this is how I've described it, and the phenomenon that I'm describing is bacterial composition, right? But if your metagenome is, or if you're studying constantly, as we mentioned, if it's an observatory and if you're constantly doing all of this, getting all of this data to observe what happens when, for example, there's contamination from the coastal area and there's just a lot of sludge coming, like untreated waters coming into the ocean, and you want to monitor that, then that would be your phenomenon to observe. And then again, what the metagenome contributes is the functional part of it. So you can do the distribution or the diversity of your community through functions. We do have it as a subvariable in the EOV spec sheet. We have functional composition, and then we also have taxonomic composition. So I guess that's how you could include your metagenomes into the observations or into this part of the specification sheets. And the contribution would be great, because that's exactly what we want. We want that data to be shared, so that when the EBVs, the biological side of it, wants to see how, for example, these things are affecting the ocean, they can through OVs, they can be interconnected. So I think it's just basically feeding the machine. That's what we want, to feed all the data into something that can coordinate an effort of observation. I hope that answered the question.
Thank you very much, Alejandra. We have another question. This is from Nuno Simos. Many of these variables need to be collected by different institutions, such as academic, government, and NGOs. What are the incentives to stimulate those institutions to adhere to this standard and share their data?
That is a very good question. Hi, Nuno. Nice to see
Yeah.
you're here. I think the incentive basically is to make use yourself of all of the information that's already in OVIS for you to use and to compare to your own data. I think it complements everything that OVIS has. If you put your data in it, you can actually complement your study with everything else that's been done about either your phenomena, your organism, or your area of the world, I think. And I agree there should be maybe more incentives to stimulate this sharing. One of the things, as I mentioned, is that I think also ODIS gives you a DOI. Once you put your data in it, it gives you a DOI. So every time somebody uses your data, they can cite your data set. And then you also get citations from using the data set. So you don't, as I said, you don't lose the ownership of the data. So I think maybe we should talk more about how this ownership of data and sharing should be established, so that the people that actually took the data can get credit for it. So, but in terms of being like specific stimuli, I think it's just about joining the network and sharing, so that you can be part of the whole system. I don't know. What do you think, Julie?
I think we've lost Julie there for a second.
Oh yeah, because she was having connection problems. I think she left. She suddenly left.
You're in the hot seat, and we've got one more question left.
Okay.
So this is from Andy Portolanton. With the 2-year period of recruitment to GOOS passing, is it possible to volunteer in providing data collection, community outreach, or software creation?
Yes, actually, it's a 2-year period, but you have renewals up to 3 times. So you can be there for 6 years. And this is very important, because I've been here for almost, it's going to be my second year now. And by the time you get to the second year is when you start understanding how everything works. I was just talking about this before the webinar with Sean, and it's very hard to get to understand how everything is frameworked through the UN and then UNESCO and GOOS and what is like the IOC committee, you know, and how everything gets put up and how the infrastructure to create this functions. So usually after 2 years, you finally understand and then they're, okay, I know what they're talking about. So now let's get to work on this side. So yes, the answer is yes, you can definitely volunteer to help with software creation. There's one person that was working, for example, with, and then once again, and I think this refers to Nuno's question. When you start using all of these frameworks, you get, and that would be an incentive. Sorry, I forgot about that. But the UN Ocean Decade programs support you sometimes. Like when you are part of these efforts, then you do get, you are, what's the word? You can apply for the UN Ocean Decade support for your programs. So because you're part of all of this. So if you create, for example, an observatory and you commit to getting all the data out for everybody else, as you see, I mean, there's a lot of centers and organizations listed here, and you can be part of that UN Ocean Decades program, and then you can get financial support for some of your projects. That's true. I forgot about that, which is very important. I don't know if there's any other type of project, but once you enter this UN Oceans Decade, then you are exposed to a lot of other institutions that are part of it, and then you can start collaborating with them, and it makes it a lot easier to find the people that you need to find to promote your work and to get your observatory or your mission out there. So yes, that's a very good incentive, and I'm sorry I didn't think about that before, but that's one. I don't know if Julie has any other, but going back to Andy's question, yes, you can volunteer and you can be part of the software creation program. There's people that dedicate their time to it, and if anyone wants to help, I'm sure they'll be happy to make you, like, to talk to you. That we are all open to talking, I think. So yeah, please check who's part of the program and contact them.
Thank you very much. Julie, do you want to add anything to what Alejandra just said?
I was just going to say, sorry, my connection dropped, but one incentive is that you'll be well cited, and the funders, whoever's funding you, likes a well cited data set, but they also like when the value from that data set is more than just the science publications that came out of it. So yeah, it's always good to report back when your data are used.
Thank you very much. I don't see any more questions in the Q&A box, and we are drawing to an end in terms of time. So I think this brings us to the end of the session. On behalf of the audience, I would like to sincerely thank both presenters again for your efforts, and to all of you, the audience, for attending and supporting another Marine Biodiversity Networking Friday. I wish all of you a pleasant weekend. Thank you. This concludes the session.
Thank you.
Thank you.
Thanks for your
Bye-bye.
attention.
Thank you, everyone.
Thank you, Sean.
Thanks for your time. Bye. Thanks, Sean.
Pleasure. Thank you so much. Really, really interesting work you guys are doing.
Thank you very much. I hope you have enjoyed it.